and there will be 2 recordings. So this is recording part 2. All right, so let me go back to, where was I? Okay. Sorry, I need to figure out where I was. Why is it not showing? Oh, there it is. Okay, technology challenged. I don't know what I was saying last. I don't know what I was saying last. Can somebody remind me?
You were discussing structural changes as a result of denaturation.
Yeah, so when you do alpha helix, I remember now, alpha helix to beta sheet, the ratio of alpha helix to beta sheet is an indication of denaturation. And it's also an indication of how the, depending on what you are looking for, if impact of processing, or if is it impact of different like ingredients that you use. If you, I don't know if you were hinting to intermediate wheatgrass flour or oat flour. I don't know your question, Radhika. Was it you?
Yeah, it was me, and I saw it for intermediate wheatgrass flour, but I assume you make a dough and then analyze it, correct?
Yes, usually you make the dough and then look at the secondary structure and try to link it to the actual rheology. And so we've done that, and I do have example to show you here as well on intermediate wheatgrass specifically. So yes, that is definitely a structural characterization that we can link to functionality. What else you can think of? Let me, so that to save some time, I'm just going to show you here. So we talked about amino acid composition and sequence, molecular size. We do that a lot, looking at molecular weight distribution that helps us to determine if we have monomers, dimers, if polymerization occurred, what type of interaction caused polymerization, if these interactions are reversible or non-reversible. So I'll give you lots of examples on those. And molecular configuration, which is basically you can look at tertiary structure, you can look at secondary structure, and primary structure by looking at the composition and sequence. And denaturation we already talked about, and then functional properties. It could be physical functional properties, which we will spend a lot of time on next time, and it could be nutritional or biological. So we have so many methods, right? And we're going to talk about a lot of different methods and what each method or group of methods tell us, and how can these methods help us understand the protein structure, or even the identification of the protein, determining its identity. We can do that by proteomics approach. We can do that by identification. If we want to identify the presence of a certain protein compared to another, we can do ELISA and Western blot. And I'll give examples on that, plenty of examples. So in order to understand, so we talked about intrinsic properties, right? So in order to understand the changes upon extrinsic factors, we have to talk about protein structure. And then we'll talk about, after we cover identification of protein structure and how it's impacted as is a native state, and then the denatured state, then we're going to explain functional properties and relate structure to function. So that's what's coming up, talking about the structure, how the structure changes upon modification or denaturation, and then how does that impact functionality. So we'll talk about structure, define functionalities, different functionality, and then relate structure to function. So that's what we're doing. So let me give you examples as to where structural monitoring the structure is important. So monitoring structural changes during processing. So sometimes when you're producing a product that's high in protein, it will impact its final texture. If it is a beverage, it might gel over time, or protein might precipitate over time. If it is a protein bar, it might harden over time. So we really want to look at what happens to the protein structure during the processing that is involved, or processing conditions that are involved during formulation. If we understand the impact of the different processing steps, we might optimize them and adjust them to reduce protein precipitation in a beverage over time, or reduce the formation of protein-protein interaction causing hardening in nutrition bars. So that's why monitoring structural changing during uh, processing or as impacted by different processing conditions help us optimize the processing conditions to get the better functionality or textural properties that we are aiming for. So understanding what's happening to the protein, did the protein denature, does the protein polymerize, does the surface property or do the surface properties change? Another example where we need to do structural characterization is when we think about valorization. Let's say you are in industry, and in industry, they also always look at adding value to their byproducts, products and byproducts. So the perfect example is the production of soy protein isolate. Originally, you have soy, and it was used here, at least in North America, for oil production and especially industrial oil production. Then somehow they discovered that the meal that was sent out for feed is particular protein characteristics that are great. So they have good nutritional quality, good functional properties, and just understanding the characteristic of the protein allowed them to produce ingredients of high value. So assessing the structure of the protein to determine its functionality or biological value is important in industry to valorize byproduct, or add, the word means adding value. Protein separation, that's a big deal. You have different sources of proteins, and every protein have different characteristics. So we need to understand the characteristics of the protein so we can get to the best process possible to retain the structure and function of the protein and not destroy it during isolation. So this is especially a hot topic now that plant proteins are becoming a big deal, and trying to figure out isolation processes that preserve the structure of the protein. So we monitor structure as we develop protein isolation protocols that are industry feasible. So that's kind of a hot topic right now. Comparison to competitor product. This is something I worked on when I went on a sabbatical with Cargill. So about 5-6 years ago, they wanted to get into the protein business, and they didn't have a protein research team, and they wanted to have this big joint venture with Puris, which is a company that actually has the plant to produce pea protein. So they wanted to get into joint venture with them and sell the protein ingredients under Cargill name. But they wanted to get into this business, but they wanted to understand their competitors. So we did a big project where we just got, 20, 25 different pea protein isolates from different competitors, and we wanted to see how they're different. Now, okay, the outcome, all of them were bad, but Cargill wanted to understand what can they tell their customers about this particular Puris protein ingredient. So we did a lot of kind of basic work understanding the structure of the protein and how it differs among these different competitor products. So that was an interesting project. Changes in functionality, and this is also another example from when I was at Cargill. So all of a sudden they were producing soy flour for a fermentation application, and they noticed a change in the fermentation yield and output. And so somehow the yeast is not able to digest the protein or consume the protein in this flour the way they did before. So, and then they brought me this this issue, and they said, well, we saw a sudden change and we don't understand why. So at that point, and I said, okay, do you have different lots? Was there anything different? And they said, no, we did the process the exact same. The only difference is we changed an equipment. We're like, okay. So do you have flour from a previous lot with the older equipment? They said, yes. So we did screening to determine the differences from before they changed the equipment, what happened to the protein after changing equipment. It seemed like they had less control on temperature time combination and ended up with having more protein polymerized, and they were because they became larger polymers, they were not as digested by the yeast as the previous flour. So a change, it's kind of like forensic work. So you want to determine where the change happened and how can you modify the process to accommodate a certain change in your equipment, for example, or anything in the processing line. And functionality enhancement is also a hot topic right now, especially with plant proteins that are very relative to animal-based proteins. They're not very functional with the exception of soy. So how can we enhance functionality? So we need to look at changing the structure of the protein. So in order to change the structure of the protein, we want to monitor how the structure is changes, right? So we process it, or we apply a functionalization technique, whether it's hydrolysis or high pressure processing or non-thermal processing or glycation. And then we look what happened to the structure, what happened to the surface properties, what happened to the molecular weight, what happened to potential interactions. Then we can relate that to enhancement in functionality. So this is, again, a very hot area of research right now, especially in the plant protein area. So this is important. It's very important because proteins don't exist in vacuum. So usually you're formulating. So you're formulating with fat, with with carbohydrates, with potentially different sources of proteins. You have different water concentrations. You have salts, you have sugars. So you want to understand if there's any structural change in the protein in the presence of other components under different processing conditions so that you can understand formulation. So now we can study emulsions in like a system, just put protein solution with oil, mix it, form an emulsion. But that's not the case for food application. We can predict emulsification properties, but it might not be the same in one application versus another. So there should be studies looking at protein structural and functional changes as impacted by the formulation or whatever it is in the formula. Again, we'll give examples throughout when we start talking about functionality and applications. Any questions? Okay. So here's how I categorize structural characterization. So we have chemical assays and we have instrument-based analyses for structural characterization. Chemical assays, basically, we use them for particular characteristics, and we use instruments for other characteristics. So for example, one of the most used tools is electrophoresis. Because what does it tell us? Sometimes simple screening with electrophoresis can tell us a lot about protein profile, about potential molecular weight of the different components, about potential polymerization, about the nature of the polymerization. So we run our protein samples, for example, in buffer, and we run them in buffer with a reducing agent. And then we can determine whether polymer, if there's a polymer, whether the polymer formed by disulfide linkages or other covalent interactions. So SDS phage is one of the most common types, but it's not the only type of electrophoresis. So we have something called SDS page because we use sodium didosyl sulfite in the buffer we use, which denatures the protein and binds strongly with the protein and gives the protein negative net charge on the surface. So when we run them on a gel, and the gel will have different pore sizes, and you run a voltage, so all of the negatively charged protein will go in one direction, from a positive charge to the negative, from a negative charge to the positive charge, and they separate only on size. So that is SDS page. Urea page, they separate based on charge to size ratio. And we use that in particular cases, for example, casein proteins. If we want to study casein proteins, we have alpha S1, alpha S2, kappa casein, beta casein. They all have very similar molecular weight. So if we run them on SDS page, we will not see separation. But if we run them on urea page, they would get separated based on charge to size ratio. Native page is you just run it in buffer without SDS, without BME, without any detergent or reagent. And we don't really see much because a lot of the proteins have large molecular weights because of quaternary structures, and they're polymerized either by hydrogen bonding or hydrophobic interactions or disulfide linkages. So you won't see any differentiation. We don't run native page usually ever at all, but some people do, just to look at the protein in its native stage, and then they do SDS or with and without BME. But we almost never do, really. So some of you are familiar with gel electrophoresis unit, and usually I bring it to the lecture and just give you kind of a show and share little demonstration. But basically nowadays you buy the cassette and the gel is already made inside of it, and it comes with different pore size, and sometimes it's a gradient, sometimes it's one percentage of acrylamide. Acrylamide is the component that you use to form the gel, and sometimes it's gradient, so it has different concentration of acrylamide across the gel, meaning you have different pore sizes across the gel. So we use different types of gel based on the proteins we're trying to separate. So the cassette is basically two plastic plates attached to each other, and inside, in the middle of it is a gel. And you have kind of a comb structure. So when you take it out, you see little wells, and that's where you load your sample in the little wells. But you don't load until you put it in a buffer in that chamber. In the chamber here, you take your gel, you put it in the buffer, then you remove the comb, and then you load a certain amount based on the size of the well. So you load your protein sample that is in SDS, and then you run the gel. You run a current, use a voltage, produce something that produces voltage, basically, and you run the gel. And then at the end of a certain period of time where you see the front line of the gel reaches the bottom, then you would usually you have the sample in a blue with the blue dye, so you see this the front, we call it the solvent front or whatever, sample front. So it goes down, and then you stop at a certain time point, and you break open the plate, and you gently take it out without breaking it, and you use a staining solution. It is, what is it? The staining solution? I'm kind of forgetting. Somebody help me. Comasi.
Imperial protein stain?
Comasi blue, right? It's Comasi. Correct? Ali, somebody?
That's correct. That's what's in our stain.
Yes, Comasi blue. Okay, so the Comasi blue basically interacts with the protein and forms this particular stain for the where the protein is on the gel. So we do the stain afterwards so that the gel becomes clear. So we wash out the excess stain. And then what we see at the end of the day are banding of the proteins that have been stained. Nothing else would stain. If you have a carbohydrate or fat, you don't want to have fat anyway because it kind of causes smearing because the fat might interact with the protein and cause some smearing. But anyway, you don't really see anything else but the protein because it's the one that is staining with the Comasi blue stain. And here, for example, in this particular lane is a soy protein isolate. So soy protein isolate doesn't have only one type of protein, it has multiple types of protein. So we're able to see the different proteins that are in there. If we have standards, we run standards, and then we know that this is beta-conglycinin and this is glycinin. And we can, for example, hydrolyze using different enzymes, and we can determine the specificity of the enzyme. For example, in this lane here, the hydrolysate, the enzyme targeted beta-conglycinin, so you don't see the banding, you see small molecular weights here. And in this lane, this enzyme targeted the glycinin, so you see beta-conglycinin and you don't see these banding, then you see lower molecular weight. So you can, for example, determine the specificity of the enzyme and what protein this enzyme targets. So it has multiple applications, looking at protein profile, looking at molecular weight distribution, looking at polymerization. I can show you later examples, and looking at hydrolysates and specificity of the enzyme. So SDS page sometimes is coupled with a reducing agent, and this is important. For example, if you look at soy protein, we'll talk about it when we covered proteins from different crops. It forms a hexamer. So you have several proteins forming a quaternary structure. This red line here represents a disulfide covalent linkage. And if I run the gel without reducing, I would get all of this kind of smearing. I don't see the monomers. But if I run the gel with a reducing agent, I'm breaking those red bonds, and then those dotted lines and dark lines are non-covalent interaction. SDS takes care of that. Then I can see my monomers. Then I see my monomers here better by running it with SDS. So in some, reducing agent. So reducing agent is added when we have a quaternary structure that is otherwise hard to see the monomers. And if we are suspecting polymerization, we add a reducing agent to determine if the polymerization between the proteins due to processing occurred due to disulfide linkages.
Pam, I have a question about that. If we use a reducing agent and produce monomers, are we underestimating higher molecular weight proteins because we're breaking them into smaller ones?
Yeah, that's why we run with and without a reducing agent. So if we're looking at extent of polymerization due to processing, you want to run it without a reducing agent, and you want to run it with a reducing agent so that you can understand the impact of if there has been disulfide linkages or not. Does that answer your question?
Yeah.
Okay, good. So isoelectric focusing is another electrophoresis technique we also use. And this technique separates based on the isoelectric point. So not based on size. So you take your proteins and then you have a gel, a strip, and then before you load your protein, you load on that strip a solution of amphilites. So amphilites basically are polymers with a lot of charges. And then you run a current. So these amphilites move along in the strip, and based on their charge, either go towards the anode or the cathode. So then you have certain charges at different positions on that strip. Then what you do is you take your protein and put it in the sample buffer, and then you load it on that strip and then run a current. So the protein moves until it reaches its isoelectric point. Then it precipitates and stops moving. Then the proteins separate based on their isoelectric point. Now, you would say, why is this important? It's important because sometimes we need to separate the protein on 2 dimensions, size and charge. So for example, oftentimes we run the strip and separate based on isoelectric point. And we take that gel strip and we place it in the comb area. And usually we order special gels that only have one big well. And we slide this strip inside that well area. And then we run the gel. We run SDS gel. What happens is then now we are separating we have our separated protein here based on isoelectric point. And then we also separate them based on size. So what looked like one band here is actually 2 proteins. And what looked as like, look at this one and this one, they're 2 different proteins, but they, oh, actually, but I'm not seeing this because here, these 2, for example, have the same molecular weight. If I ran them directly on SDS page without separation here, they would look as one protein. So this is an example where, where we ran one-dimensional gel and two-dimensional gel. So here you have a soy protein isolate. We take that one protein, soy protein isolate, just that one here, and we run it on a gel. If you look here, how many different spots you see? Several many spots you see that shows up here as just one band. This tells me that this glycinin, acidic subunit of glycinin, we'll learn about it later, actually has several variants with different, similar molecular weight, but potentially different homology, different sequence, different amino acid sequence. So that is telling. One sample can give us the different proteins that otherwise looked as if they are one protein.
Pam, I have a question.
Yes.
So instead of doing urea phage for MCC or like a micellar casein, can we do an SDS and do a 2-dimensional one, like precipitating with the PI too?
Yes, you can. You can, but if you can separate them using urea for a particular purpose, so you can do 2 in 1 rather than having to do 2D.
Okay.
I mean, always 2D is better information, but the problem with 2D is you only run one sample at a time in one gel, because you ran your you run your SPI, for example, soy protein isolate. In a one-dimensional gel, you have multiple wells and you can run multiple samples. But in a 2-dimensional gel, you are running one sample at a time. Depends on the purpose. Sometimes you do it if it tells you what you want, and sometimes you choose not to do it.
Okay.
It's case by case. Now, this is an example where running a 2-dimensional gel told us a little bit about a new type of protein, which is a camelina protein. If you see the proteins, they're mostly in the basic area of the gel. So you have a range between pH 3 to pH 10. So that you see them concentrated here in the neutral to above the neutral. So this tells me that this protein is mostly basic protein. And if we want to extract it using pH, alkaline extraction, we really have to have a higher than the isoelectric point where the protein carries charge. Then you are extracting the protein at pH 10, 11, or 12 sometimes, which is really detrimental to the protein quality, which is, it could cause permanent denaturation and polymerization. But this is the characteristic of the protein, and alkaline extraction means we really need to have to do really high pH extraction. Capillary electrophoresis, it's more quantitative than the regular gel electrophoresis. Here you can have a capillary tube, and in the capillary tube, you can have your gel matrix. You can have SDS type of gel, or you can have an isoelectric point type of gel. And then your samples can be separated based on isoelectric point, or based on charge, or based on size as well. But the benefit of this, it's used for quantification. So you can put detectors similar to HPLC detectors, and you can actually separate and quantitate as well. Okay, so surface properties. So we do that a lot in the lab because we want to know what happens to the surface properties once we either denature the protein or subject it to processing that might cause denaturation, or maybe modify it in a way that might change its surface properties. Surface properties are important because if you want to form a gel, surface property interaction with water, interaction with protein is important. If you want to form an emulsion, the balance between surface hydrophobicity and surface hydrophilicity is important. That's why we need to look at the net charge on the surface, as well as the hydrophobicity on the surface, which can explain why one protein is a better emulsifier than another. Free sulfide group is important in certain application. If we're trying to form a stable gel or a stable dough system, we want to have more disulfide linkages. And we look at different proteins and determine, well, this protein has more sulfide group on the surface, or this processing causes more disulfide sulfide groups on the surface. So there's more potential to polymerize. Another one we do sometimes in the lab is called looking at amine groups. And we use that by doing an OPA acid of aldehyde reagent that reacts with the amine groups of the protein. So sometimes we do it to see if processing caused blockage of or degradation of lysine. Sometimes we do it to monitor the hydrolysis. The more peptides are broken, the more amine groups are released, then the more reaction with OPA there is. So it has different applications that we use in the lab. Sometimes we want to polymerize the protein by transglutaminase enzyme, so which involves a lysine, an amine group, and a carboxyamide group of a glutamine. So we can determine blockage of amine based on the use of the enzyme. So it has several applications that we use in the lab for structure characterization. Any questions? Okay. And don't worry, there's a lot of information, but all will come together when I start giving you examples, linking structure to function. But now I'm just giving you an overview of the different structure characterizations we do. Okay, looking at structural deviation, changes in the molecular configuration. So looking at denaturation of the tertiary structure, differential scanning calorimetry. So basically we look at what temperature does the protein denatures, that particular protein denatures, which is important to know so that we know impact of thermal processing on the denaturation of the protein. And it's also important to tell us whether or not the protein is already denatured in a certain ingredient, or if a certain processing caused partial denaturation compared to another processing that might cause complete denaturation. So it gives us information such as enthalpy and change in enthalpy. And I'll show you the graph later on. Protein secondary structure by FDIR, which we already brought up. It also, like the ratio of alpha helix to beta sheet, is an indication of denaturation at the secondary level, secondary structural level. Also, presence of beta sheet, for example, is important for stable dough. In the example that we just talked about earlier, when you have more beta sheet and then formation of beta turn in the presence of moisture, that gives you good rheological properties. So this is something we look at. And we sometimes look at protein tertiary structure using Raman, which is a light scattering type of detection, also based on infrared, based on IR radiation. And it gives a lot of information about molecular interactions and shifts and changes in the molecular structure due to processing, whether it is heat or pH or what have you. I'll give you examples later. So this is a DSC, differential scanning colorimetry thermogram. So the thermogram is basically when you run your sample, you put the sample in a compartment, sample compartment, you seal it, and then you apply a heat flow over time. So you have a method where you determine the rate of heat that you want to apply over a certain period of time. And then you get two important things out of here is the temperature of denaturation. So the uptake of heat, whenever you have an uptake of heat, we get something we call the endothermic peak. So the peak you see here, the little peaks here, they're called endothermic peak. So if the protein is in its native form and then you apply heat, it's going to uptake heat and denature. Then this endothermic peak is going to tell me at which temperature this happened and how much heat it took. So the change in enthalpy. So the change, the amount of heat needed to cause a conformational change. So what you're showing here is there is a sample that shows nothing. When you don't see anything, what does that mean? Can one of you tell me when you don't see any endothermic peak, what does that mean?
It's already denatured.
Already denatured. Goodbye. That's a commercial soy protein isolate. We compared it to soy protein isolate we produce in the lab. We see two endothermic peaks for the main components, glycine and beta-conglycinin, but we don't see any endothermic peaks for a commercial sample. That tells me it's completely denatured. So it's one of the characteristics we can differentiate a protein that is in the native form versus a protein that got processing that caused denaturation. So FDIR, without a lot of going through specifics, I spent a whole lecture in analysis on FDIR, but here I just want to remind you this is based on IR radiation absorption. And when the molecule absorbs IR radiation, you have vibrational changes in the vibrational energy. So basically you have stretching and bending of the bonds, and these are specific to the type of functional groups and the bonds that are there. So it tells us information about the type of bonds. So here's an example of, we just talked about intermediate wheatgrass, for example, versus hard red wheat. So wheat is wheat, obviously, and the wheatgrass is a grain similar to wheat, but doesn't have the same functionality. It doesn't form as viscoelastic dough as the wheat does. So we wanted to dive into it and look at what at the molecular level, why don't we get a viscoelastic dough? So what we did here is basically looking at the protein secondary structure. So the amide 1 is a region in the protein that gives you information about the protein secondary structure. That's all you need to know for now. And it is in between the 1600 to the 1700 wave numbers. And you can deconvolute the peak. So there is a mathematical equation that you would do in the software that you deconvolute the peak, and it gives you the distribution of beta sheet, random coil, alpha helix, and beta turn. And we ran this analysis on doughs produced by wheat and wheatgrass, and we saw differences in the distribution of beta sheet, alpha helix, random coil, and beta turn that explain differences in rheology. This is a more recent study that we have done in the lab, looking at differences between a commercial pea protein isolate and a pea protein isolate we produce in the lab, and also protein isolates that we have modified with non-thermal type of processing called plasma species. So, but I don't want you to worry about a lot of different numbers here, but I want you to look at commercial versus the one that we produce under optimized method to preserve the structure. So you see a major difference in random coil. That's an indication of denaturation. You see a lower alpha helix to beta sheet ratio, and that's also an indication of denaturation. So alpha helix, when you heat, basically it converts either to beta sheet or random coil. We don't see much difference in beta turn, but this helps us understand how our way of producing the protein preserve the structure compared to a commercial source, and also how different treatments impact as well our structure. I'll spend a few minutes on Raman. So we've done work with the Raman spectroscopy and mostly surface enhanced Raman spectroscopy. Again, there is a sheet that describes the Raman surface enhanced Raman spectroscopy on Canvas, but basically it is based on light scattering. So the IR radiation hits the sample, and then the sample scatters light and carries characteristics specific to the structure of whatever material we're studying. This is, for example, a whey protein. So the disadvantage of Raman is you have very, very low signal. So especially if it's in liquid, so if it's a solution, you have very, very low signal. But the advantage of Raman over FDIR is you don't have interference of water interference. Water does not absorb in the IR region. So we can, what does not scatter light in the IR region. So we can measure solutions, not only powder. So that's an advantage. However, at low concentrations, you have very low responses. But with surface enhanced Raman spectroscopy, you can use structures, nanoparticles, such as the silver nanoparticle, that where the whey protein can adhere to the surface particle, and with that increase the signal. And it's easy to run. Usually you just have your plate with your nanoparticles, and then you would spot at different places, and you have a little microscope, and you would project the light, and then you measure the scattering of light. So an application for that. So here, for example, we were looking at structural change when we modify whey protein. So we modified whey protein by covalently linking a carbohydrate to it. And we found that when we covalently linked the carbohydrate to it, the solubility enhanced. And the structure remained stable even after heating. But here, what I would like just to see is what with Raman, you can look at different bonding and different characteristics of the protein. For example, one important characteristic we looked at, or bond, is the bond between carbon and nitrogen, which is the characteristic bond of the Maillard reaction bond, where a carbonyl group reacts with a amine group. So we were able for the first time to basically detect a new peak at 983 that is not in the actual sample. So we were able to detect this peak at this particular wavelength which confirmed the formation of Maillard reaction. We were also able to see differences in SS linkages, in the amide groups, beta sheet, for example, differences in the ionization status between a carboxyl group that is not ionized and a carboxyl group that is ionized. So we were able to explain enhancement in solubility by looking at specific functional groups and bonding and shifts using surface enhanced Raman spectroscopy. So we look at protein aggregation and molecular weight by size exclusion chromatography. We talked about that. We can use different detectors, but if we use a specific detector that is based on light scattering, multi-angle light scattering, we can get accurate molecular weights without having to do standards. But it's a very expensive detector. Sometimes we just run our regular UV detectors and use standards to look at the formation of soluble aggregates, understand how the aggregates formed. We run the sample under different conditions, buffer with SDS, with BME, and try to understand what type of interactions happened within the protein. Microscopy, SEM, scanning electron microscopy, we use to look at morphology. There's also the confocal laser microscopy, CLSM, scanning microscopy. I don't have it here, but also it kind of helps looking at distribution of protein, let's say an oil and carbohydrate in a plant-based cheese, for example, which I'll show you images later on when I show you some examples about structure-function relationship. So microscopy can help us look at aggregation by looking at morphology, but also can help us look at a distribution of the different components. Especially with CLSM, we can do staining and look at different components within a matrix. All right, I will spend 2 minutes. I will stop at 4:50, but here is size exclusion chromatography that helps us, for in this particular case, look at protein types in, again, wheat versus this intermediate wheatgrass type of grain, where again, we look at the distribution of the different gluten forming proteins, and to understand why the wheat forms a better dough than intermediate wheatgrass, because we have more high molecular weight polymeric proteins than wheat than in intermediate wheatgrass. So size exclusion chromatography helped us determine the ratio of these different proteins, trying to understand why one is more functional than the other. Here's another example that we used the size exclusion chromatography for, and this is recent. We looked at, again, formation of soluble aggregates versus the monomers in pea protein in commercial versus native versus treated proteins by cold plasma. So we can see differences in formation of soluble aggregates, which could potentially give better gelation properties. So if you have soluble aggregates, you have better tendency to form gels than only if you have small molecular weight monomers versus large molecular weight insolubles. So the size exclusion chromatography helped us look at that distribution and understand why one protein formed a better gel than another. This is another example, final example on size exclusion chromatography that we use to look at specificity of an enzyme towards whey protein. So here peak 4 is beta-lactoglobulin and peak 5 is alpha-lactalbumin. When we used an enzyme, a specific microbial enzyme, it targeted alpha-lactalbumin and produced smaller molecular weights here, peptides. Whereas when we use trypsin, for example, trypsin targeted peak 4 here, which is beta-lactoglobulin, and did not hydrolyze much the alpha-lactalbumin. From looking at the peaks, we can see different specificity, but we can also analyze and get molecular weight distribution. We can get peak intensity and determine changes based on the different enzyme treatments, higher degree of hydrolysis 8, for example, compared to 5, or 12 compared to 8, and different enzymes. So it tells a lot of information that helps us understand certain characteristics of the protein. Okay, I'm going to stop here and we'll continue next time. All right, thank you guys. I will see you. You will get 2 recordings from today's lecture. Don't forget that. I'll post that on Canvas shortly. Bye, you guys.